Hello, and welcome to a new semester of Immunology. This video series is part of a 400-level course in immunology offered by the Department of Cell Biology and Neuroscience at Rutgers University, but you're very welcome to join us and learn with us wherever you're watching this video from around the world. In this first lecture in this new series, we're going to cover some of the very basic principles of the immune system, including some of the major divisions in the way that the immune system is organized. And as you can see here, that will involve a very preliminary discussion of innate versus adaptive immunity. But before we go there, I'd like to talk about some basic principles of molecular biology that I think will guide our thinking about the way the immune system operates, especially early in the course. So, a major principle of molecular biology that I think is absolutely essential for understanding the immune system is this sentence: structure confers function. A way that one of my previous undergraduate professors put this was a different saying, which is, the shape of the glob determines the job, referring to the globular shape of most protein molecules. The idea here is that the shape of almost any biological molecule is almost everything in determining the types of things that that molecule is able to accomplish biologically. And so an example you can see here in this cartoon is that individual substrates all have a unique molecular shape, and that's going to determine a unique partner enzyme that they bind to and will be metabolized by as a result. And so this is a pretty familiar concept to many of us who study molecular biology, that there are molecules whose shape is complementary to other shapes and that determines how they interact. But we could dig a bit deeper and think about the way that shape determines some other fundamental features of protein interaction and molecular interaction for that matter. If we focus on proteins for now, the individual shapes of two molecules that are interacting, or of any molecule on its own terms even, are going to determine these four things that you see listed here. We'll begin talking about specificity, meaning which molecules will any given protein bind to. And this is probably most well illustrated by the image here, where shapes tend to be complementary to each other, and so a given substrate or a given ligand will only bind to another molecule if its shape is complementary in some way. They fit together like a lock and a key. However, beyond the specificity of a given interaction, the shape of a protein or its structure will also determine things like its affinity. How strong is that association? So determining already that two molecules interact in a specific manner, how strongly and how long-lasting do they bind? We refer to that idea as the affinity of their interaction. Beyond affinity, the shape of a molecule is going to determine what biological function it performs, and this is known as its activity. So if it's a substrate that might be in an enzyme pair, that might be some kind of enzymatic reaction. It may be a receptor-ligand pair that transduces some intracellular signaling cascade. But ultimately, there are lots of different biological activities that molecules can perform, and they again are primarily determined by molecular structure or shape. Finally, the shape, the structure of a molecule determines its stability or its resistance to degradation. And so particular conformations of proteins and particular shapes render them more or less susceptible to things like low pH or high temperature in terms of how well they can maintain their tertiary structure even in the presence of some kind of environmental stressor or perhaps some biological stressor like a protease enzyme. And how stable a molecule is very important, particularly in the context of biological reactions, and immune reactions are no exception to this. So, for example, there are some immunological molecules like immunoglobulins or antibodies, which are highly stable, and their interactions with their targets last for a very long time because they are highly stable molecules. Whereas there are others, like some certain cytokines or chemical messengers of the immune system, which are very short-lived, and that's good because we only want them to be active for a very short window of time. Therefore, they tend to be less stable. So these four sort of themes that arise from the structure of proteins, we'll see them come up over and over again as we think about different proteins of the immune system and how they interact with their various targets. If we think about the targets of the immune system, your mind may immediately go to pathogens, and that would be correct, because when we think about the immune system, probably the most fundamental job that it performs is to fight infection. Although, as we'll see this semester, the immune system actually does many things beyond that. But let's focus with the basics, as we said in this first video, and think about what the immune system needs to interact with in order to accomplish its role, fighting infection and protecting the host. A major thing that I want to highlight immediately are that there are a lot of different types of infections that you might encounter in nature over the course of your life. As you can see here, there are 4 major types of pathogens that we encounter: viruses, bacteria, funguses, and parasites. And each of these are very genetically and structurally distinct from one another. That is to say that each of these types of pathogens all harbor their own unique types of shapes in terms of the molecules that they express on their surfaces that are available for our immune system to detect or interact with. This broad variety of shape across pathogenic phyla and across their evolution is a challenge for our immune system, because we need to be able to functionally interact with and recognize a very, very diverse set of molecular shapes. And that is, in fact, one of the major accomplishments of our immune system through our own evolution, is evolving the ability to distinguish shapes and know that they belong to pathogens that we then need to mount some sort of response against. And so the shapes that are associated with pathogens have a particular name. We call them pathogen-associated molecular patterns, or PAMPs, P-A-M-P. These patterns have to do with individual sort of amino acid sequences, typically, that together fold into some sort of recognizable and reproducible shape that we then can interact with our own set of host proteins. And these, as a broad class, are known as pattern recognition receptors, or PRRs. And through evolution, we have developed a pretty broad class of pattern recognition receptors, or PRRs, that are able to individually distinguish unique types of PAMPs, pathogen-associated molecular patterns. So as you can see here in this example, this receptor that we have here drawn in purple is recognizing a particular PAMP that we can imagine as a circle in blue, whereas a different pattern recognition receptor is going to recognize something that we might imagine being more triangular or more square. And if you think of each of these shapes maybe as arising from a different pathogen, maybe the blue is from a virus and the orange triangle is from a bacterium, you can get the idea that we can express different unique receptors, and the unique shapes that those receptors are able to interact with is a molecular way that we can distinguish individual pathogens from each other, and therefore begin to mount specialized immune responses to each. So what are some of the shapes that help us determine whether or not we are encountering an infection? How can we use shape to distinguish a pathogen? Well, there are many types of pathogen-associated molecular patterns, or PAMPs. I'm going to give you a simplified version of a very classic one here on this slide. If we think about the fact that we need to distinguish shapes, we can exploit the fact that proteins and nucleic acids associated with pathogens, by definition, have to be different somehow from our own host nucleic acids and proteins. We are not viruses, viruses are not human, and therefore there are going to be differences in these fundamental macromolecules between species. And these fundamental differences in these macromolecules are going to express themselves as differences in structure and in shape of these given molecules. So you can see here, for example, that we express pattern recognition receptors that are capable of distinguishing the 5' cap of viral RNA from the 5' cap of our own human RNAs. And again, we're oversimplifying. We can imagine that the 5' cap of a viral RNA is somehow more circular and therefore is more complementary to this kind of round pattern recognition receptor that we have, which is then incapable of recognizing the more square, quote unquote, 5' cap of the human RNA. And this is good because we need to be able to say, hey, there's viral RNA here, we have an infection, we need to do something, but then not recognize human RNA. We need to be ignorant of our own RNA. We don't want to recognize it with our immune system because we don't want to sort of mount a response that would be inflammatory or somehow harmful against our own self molecules. And this is where we get the idea of this slide, the title of this slide: the immune system must distinguish self versus non-self. That is essentially what the idea of pattern recognition is all about. How can we discriminate, identify molecular shapes that belong to non-self entities like pathogens, and also understand which shapes belong to us, belong to the host, and are therefore self molecules or self shapes? Pattern recognition receptors are one mechanism that the immune system uses to accomplish this discrimination. And this is important because while it's important that we recognize non-self molecules so we can attack them, so we can control them with our immune responses, it's very important that we do not attack, we do not mount immune responses against self molecules. And in fact, the way this works is that we have a particular term for this, our inability to recognize self molecules with our immune system, and we call this immunological tolerance. We want to tolerate our own molecules so that we can maintain homeostasis. And in individuals, in organisms that, for whatever reason, through a genetic mutation or some change over the course of their life, begin to recognize self molecules as foreign through binding through some kind of pattern recognition receptor, for example, this is the basis of what we call autoimmunity. Auto meaning self. Some immune response against some self molecule. This is when we have a breakdown of immunological tolerance and we begin to mount immune responses against self. And so we'll learn later in the semester about some of the processes that lead us to begin to recognize self molecules as foreign, and this is the source of many autoimmune diseases. So these are some basic principles, as I said, and so far we focused on pattern recognition receptors as a means by which we recognize self versus non-self. However, the immune system has other tools at its disposal that allows it to distinguish self versus non-self. Another one that we can learn about is the recognition of something that we call antigen by antigen receptors. And a particular type of antigen receptor that you've probably heard of before are called antibodies. And so these are two terms which are going to be super essential to learn as you sort of begin your journey of learning about immunology. And the interaction of antigens and antibodies looks something like this. A pathogen, in this case a virus, is going to express some protein on its surface which will be suitable for recognition by our immune system. This is similar to a PAMP, but it's not exactly the same. That's because a antigen is also some sort of non-self entity that we can recognize, but it's one that we sort of learn to recognize and we adapt to, as opposed to something that we're born being able to recognize. And I'll say more on that point in just a couple of slides. But for now, antigen is a type of non-self entity that is different than a PAMP, and it's a piece of a protein in particular that's taken from some kind of pathogen, and that we can use that piece of protein to stimulate pathogen-specific adaptive immunity, something that we sort of adapt to over the course of our life as we're exposed to it. And if you think about the etymology of this word, you have anti, meaning against, and the suffix gen, meaning to generate or to start something. So it's some piece of a protein that can generate an anti-against response against itself, essentially. And antigen can be abbreviated with the letters AG. Antigen can be recognized by a few different parts of the adaptive immune system. Here you can see, again, one of the most famous examples of something that recognizes antigen, and that is an antibody, or a body meaning some sort of entity associated with the host that can perform that anti-response. It can recognize the antigen. And antibodies are unique proteins produced by a cell called a B cell, and they bind with very high specificity to an individual antigen and perform a variety of functions that then help to eliminate the pathogen that antigen is associated with. So I've used this term adapt and adaptive as part of the immune response. So I think it's time now to maybe dig further into the different arms of the immune response that some of these different ways of recognizing non-self derive from. So I want you sort of to organize the material that we're going to learn this semester into sort of two major arms of the immune system that operate at different time scales and with different levels of specificity and arise from some different types of genetics within our own host cells. But for now, let's kind of start with the most basic and broad overview, thinking about categorizing the immune response into both innate immunity and adaptive immunity. A major difference that is at the very top of this chart is that innate immunity occurs very quickly. It's very fast. It happens on the course of minutes to hours. Whereas the adaptive immune response is slower. It takes many days to really get going. In terms of the specificity of these responses, a major difference between the two is that innate immunity is limited and fixed. We express a limited set of things like pattern recognition receptors, which are very good at detecting non-self entities, but they can only cover a limited amount of non-self molecules. They don't have a limitless possibility in terms of the different things that they can recognize. Whereas our adaptive immunity adapts. It's able to sort of learn, if you will, over time and recognize a truly staggering, broad array of different types of pathogens. And so for that reason, we think of it as more diverse and more dynamic. In terms of the how strongly this response occurs upon individual infection events, the response to repeat infection with our innate immune response remains the same each time. So every time you have an infection, you basically have an identical innate immune response to it. And you can see this pictured in the graph below, which we'll come to in just a second. And if we compare that to our adaptive immune response, adaptive immunity actually gets progressively better and better the more times we are infected with a given pathogen. So let's dig into this graph because it illustrates some of the points that we just went over here. If we sort of graph an immune response and we put the magnitude or the strength of the response on the y-axis and just generic time on the x-axis, just as time passes following an initial infection event. If we think of our innate immune response occurring after that initial infection, you can see that it goes up relatively quickly in comparison to the adaptive immune response, which is drawn in red. It comes up first, and it peaks kind of at a low level, and then it resolves, and it goes back to zero over time. And in comparison to the adaptive immune response, you can see that it's coming up later. It takes longer to get started. But in terms of its overall magnitude, it reaches a greater height. It's actually stronger even on that first infection. And it's going to resolve a little bit later compared to our initial innate immune response. But notice upon reinfection, when we get exposed to the same pathogen for a second or third or fourth time following the initial infection event, notice that the magnitude and the timing of the innate immune response is essentially identical to what it was the first time we saw the pathogen. But now the adaptive response, it starts much faster, almost synchronously with the innate immune response. It peaks even higher than the already high peak it had the first time, and then it lasts even longer as well. This is because the adaptive immune response adapts over time. It gets progressively better. And I think that's captured quite nicely in this pictorial representation of these features. Another thing on this slide that we will cover in more detail is that all of these differences in the innate and the adaptive immune response arise from the fact that they are mediated by different types of cells and different types of tissues. The innate immune response is primarily mediated by barrier tissues, like the epithelial cells lining the gastrointestinal tract or the reproductive tracts, as well as cells called phagocytes, which are very good at eating pathogens, whereas the adaptive immune response is mediated primarily by cells known as T cells and B cells, and particular molecules that are made by B cells called antibodies. And we'll dig into each of these cell types and the things that they make in greater detail in future lectures. But another thing that I want to cover, which really is a fundamental reason why all of these differences in our innate and adaptive immune response, why they differ so much, is that innate and adaptive immunity arise from really distinct genetic programs. So this slide sort of covers a few different vocabulary terms that we've covered thus far and helps us sort of distinguish them. I want to say that in general, innate immunity relies on the recognition of non-self entities, or PAMPs, pathogen-associated molecular patterns, by pattern recognition receptors. And you can think of PAMP-PRR interactions as really the source of most innate immune responses. And pattern recognition receptors that we express on the host side are encoded in the germline by stable genes that are very highly conserved throughout metazoan evolution. What that means is that we have inherited, not only from our parents, but from our deep ancestors in evolutionary time, a set of receptors that can recognize pathogens, and that we've been using them basically since we were single-celled organisms. And we see the existence of pattern recognition receptors across a broad taxonomic range. So plants have pattern recognition receptors, insects have types of pattern recognition receptors, and we have needed them basically throughout our entire evolutionary history because we have been co-evolving with pathogens. We've needed to be able to detect them, and therefore we inherit genes that code these receptors in our germline DNA, meaning in all the cells of our body, essentially. And these pattern recognition receptors are indeed expressed by all nucleated cells, basically every cell type in your body. And if you're a mammal, this includes basically all cells except red blood cells, which do not have nuclei. In humans. We can contrast this with the way that we recognize non-self in the adaptive immune system. Adaptive immunity, instead of relying on PRRs to detect PAMPs, instead relies on antigen receptors to recognize antigens. And so anytime we're talking about antigens and antigen receptors, we're still talking about the recognition of non-self, but we're talking about the adaptive immune response. And adaptive immunity can recognize a much broader class of potential non-self antigens. There are many more antigens that we recognize than PAMPs, for example, because we make our antigen receptors with specially recombined genes that we sort of make in-house. We don't inherit them necessarily from our ancestors, but within our own bodies, we can recombine a set of gene segments to make a much broader class of antigen receptors that can recognize many, many unique and distinct potential antigens. And this allows us to recognize all the billions and trillions of particular unique proteins associated with all the possible viruses, bacteria, parasites, and so on that we might encounter in nature. We'll unpack that a lot further in further lectures, but what I want to really stress here is that this is a process. Somatic recombination, where we make antigen receptors, is something that happens uniquely only in B cells and T cells, and therefore they are the main sources of our adaptive immune response. Okay, so we've covered that there are different ways that we recognize self versus non-self in these two arms of the immune response, and we see that different cell types can mediate these two processes. So let's finish this first lecture just by covering some of the major functions that are mediated by innate versus adaptive immunity. Innate immunity, its first job is to, and remember that it responds quickly, so it's really as our first line of defense, is to perform the initial detection of infection, and it's going to do so through the recognition of PAMPs by pattern recognition receptors, or PRRs. Once it detects infection, it has some very important early functions that help to sort of control infection in its early stages. That includes phagocytosis, or cellular eating of pathogens. So as we'll see, the innate immune response is mediated by a family of cells, many of which are very good phagocytes. They're very good at eating things like bacteria. And that is very helpful to us initially to control the numbers of pathogens once they first establish infection in our body. Beyond phagocytosis, many innate immune cells can secrete soluble mediators, including molecules like cytokines and chemokines, which are a chemical messenger of the immune system that we'll talk more about, that sort of modulate the immune responses of other cells, as well as cytotoxic effector molecules, things that are directly antimicrobial, can actually go and kill bacteria, kill parasites, kill funguses directly through some direct toxic mechanism. And so this secretion of soluble mediators also happens very early by particular cells of our innate immune response. And then finally, innate immune cells also sort of serve as a bridge between these early stages of innate immunity with later stages of adaptive immunity, because they do something called antigen presentation. They present antigen to T cells in particular and help them to recognize non-self and become fully activated to initiate an adaptive immune response. So even though we think of many phagocytes mainly as part of innate immunity, they have a role to play in priming or generating adaptive immune responses through the process of antigen presentation. And again, much more about that to come. Let's then think about major functions of the adaptive immune system. Well, we've talked about a few cells that belong to the adaptive immune response. That includes B cells, which, as we've said, produce antibodies. And antibodies themselves have a variety of functions that allow them to fight infection. They can bind to pathogens, as you can see here with this bacterium. Sometimes just binding to a pathogen just sort of interferes with its ability to do what it wants to do to mediate some infectious process. But antibodies also target the things that they stick to for killing by other components of the immune response. And so antibodies are very effective antimicrobial molecules produced by B cells. T cells actually come in multiple flavors. Again, more on that to come. But for now, you can remember that there are two major, in the broadest possible classification of T cells, we can think of two major subsets. There are T cells called T helper cells, or TH, and they primarily modulate the immune response through the secretion of soluble mediators called cytokines, and these cytokines support the function of B cells and other immune cells. Helper T cells help other cells to control the immune response. In contrast, cytotoxic T cells, or T subscript C, as you can see here, their main function, as their name suggests, is to be directly cytotoxic, meaning to kill a cell, cytotoxicity. And so they are capable of recognizing cells that have been infected and killing them, and thereby killing the infected cell, they remove it as a niche for that pathogen to replicate itself and to further spread throughout the body. So cytotoxic T cells are especially important for controlling intracellular pathogenic infections by viruses, for example. Finally, both B and T cells also, they serve short-term functions like that we've just covered, but they uniquely are capable of producing long-lived memory cells, which can persist in our body, sometimes even for life, and they are the source of those progressively better and progressively faster immune responses that we can have to reinfection. We have memory cells which become poised to recognize a pathogen on subsequent infection events and sort of mount fresh effector functions, fresh responses to those pathogens using antibodies, using cytotoxic approaches. And so we will see that immunological memory is a unique feature of the adaptive immune response, which makes it particularly powerful. Okay, so let's summarize what we've covered in this first video. I have stressed that molecular structure determines molecular function, and the structure-function relationships of molecules are really important for facilitating targeted immune responses. And so we learned that both PAMPs and antigens are features of pathogens that mark them as non-self so we can recognize them, and that we have sensor molecules like pattern recognition receptors and antigen receptors that allow us to distinguish non-self, but importantly, when things are going right, cannot recognize self and therefore do not initiate autoimmunity. We saw that the immune response has two major phases, including innate and adaptive immunity, and we learned that innate immunity is relatively fast, but is somewhat limited in comparison to adaptive immunity, which is slower but much more dynamic. Finally, we saw that innate and adaptive immunity arise from distinct genetic programs. Innate immunity is very ancient and relies on receptors that are hard-coded into the germline DNA and are highly conserved across metazoan evolution, whereas adaptive immunity is unique to vertebrates and relies on a very unique genetic event that occurs in B cells and T cells called somatic recombination. Okay, that covers our very first video introducing the immune system. I'll see you in the next video where we'll dig a bit deeper into the process of antigen presentation.